All right, welcome. Today, we are going to step away from the nice, neat world of structured databases and dive headfirst into the wild territory of bioinformatics data, those huge, sprawling text files. We're going to unlock the power of two command line superheroes, Bioawk and Sed, to ask questions and even edit this data, all on the fly. Let's get into it.
So I'm willing to bet this is a question you've had. If you come from a background with SQL where you can just query clean tables, looking at a raw genomics file can feel, well, a little chaotic. It's a super common problem, but thankfully there's an incredibly powerful way to tackle it. Okay, look at this. On the left, you've got a perfectly logical SQL query. We all know what that does. On the right, a multi-line FASTA record. They're both trying to do a similar thing, right? Get a specific piece of genetic info. But the formats are just worlds apart. Now, loading everything into a database is one way to go, but with gigantic files, it can be really slow. So in bioinformatics, we often treat data as a stream. We process it as it flows past us, which is way, way faster. So, if a database isn't always the right tool for the job, how do you query these files? Well, the answer is to use a specialized text processor. And the first one we're going to meet is Bioawk. This is where Bioawk becomes your new best friend. It's built on top of a legendary tool called Awk, but you can think of it as Awk that went and got a PhD in biology. It's a text processor that already understands the structure of the most common bioinformatics file formats, which just takes all the painful guesswork out of trying to parse them yourself. And this is the real magic right here. Bioawk gets all these formats straight out of the box. That little C flag is basically like telling a database, hey, here's the schema for the file I'm giving you. When you say C fastx, Bioawk instantly knows how to read FASTA or FASTQ files and separate things like names, sequences, and quality scores. It's amazing. No more writing your own complicated parsers. Okay, so that's the theory. But let's get practical. How would we actually use Bioawk to do something useful? Like, say, grab the name and the length of every single sequence in a file. So we're going to build this command piece by piece. First up, we just call Bioawk and give it that -c fastx flag. We're just telling it, hey, get ready, the file I'm about to send your way is in FASTA or FASTQ format so you know what to do. That's step one. All right, now for the action. This part in the curly braces is where we tell Bioawk what we want it to do for every single record it finds. Here, we're using two special variables that Bioawk creates for us, $name for the sequence identifier and $seq for the actual sequence. We just say print the name and then print the length of the sequence. It's just like selecting columns from a database table. And here it is, the whole shebang. A single, beautiful command that reads a compressed file, understands its format, does a calculation, and spits out this perfectly clean tabular data. We didn't have to import anything into a database. We didn't have to write some long, complicated script. This is the power of stream processing in a nutshell. Okay, that's awesome for pulling data out. But what if I need to make a few little changes? You know, like cleaning up a messy header or reformatting a column before I pass it on to another program. For that, we bring in our second tool, Sed, which stands for Stream Editor. You can think of Sed as a super fast surgical find and replace that works on your data as it's flowing by. It is perfect for making those quick, precise little tweaks right in the middle of a pipeline. A classic example is cleaning up headers. Let's say Bioawk spits out a header that says something like gene_XYZ/chromosome1/human. But the next tool you're using is picky and it only wants gene_XYZ, no problem. You just pipe that output into Sed and tell it to delete everything after that pipe symbol. It reformats the data on the fly, making it perfect for whatever comes next. So we have Bioawk to extract and Sed to edit. But the real game changer, the thing that makes this whole approach so powerful, is when we ask, can we chain them together? Can we build a whole multi-step workflow? And the answer is a resounding yes. That question really gets to the core of the whole Unix philosophy, which is captured perfectly in this quote. The idea is simple. Create small tools that each do one thing really, really well. Then connect them using pipes to build powerful, custom workflows. It's like building with Legos. So this is what it looks like. Data flows from a compressed file. It immediately hits Bioawk, which filters out only the reads we care about. That filtered data then flows directly into Sed, which tidies up the names.
And only then, at the very end, is the final clean result written to a new file. At no point did we have to save a bunch of temporary files. It's all happening in memory in one continuous stream.
So why do we work this way? What's the big deal? Well, first, it is blazing fast. Processing in memory is just orders of magnitude faster than writing to a disk. Second, it's efficient. You can process a terabyte-sized file on your laptop because you're only ever looking at one line at a time. And finally, your work becomes modular and reproducible. The whole analysis is just a single line of code that you can save, share, and run again and again perfectly every time. You see, this whole approach is more than just learning a few commands. It's a fundamental shift in how you think about your data. You stop seeing it as a static file that you have to load, and you start seeing it as a dynamic stream that you can shape, filter, and transform as it flows. So the real question is, now that you have these tools, how will you ship your data?
And that's our look at Bioawk and Sed. If you found this useful, do me a favor and share it with a colleague who might also get a kick out of it. And of course, make sure to subscribe for more deep dives. Thanks so much for tuning in.